Hello, my name is Stefan Lalonde. I'm from the Laboratory Geosciences Océan in Brest, and it's my pleasure to talk to you today about oxygenation of the early Earth. Before we get started, I'd just like to talk about what do we mean by early Earth. Here you have the geological timescale according to the Geological Society of America. And what you can see here is that you have the Cenozoic, the Mesozoic, and the Paleozoic defined in great detail, but this only represents about 1/8 of the sedimentary record. And on the far right, you see the entire Precambrian, which represents about 7/8 of Earth's history, compressed into one column. But so this is the geological timescale, not actually to scale. Today I'm going to be talking largely about the Precambrian, the vast majority of Earth's history where a lot of these important events in Earth's oxidation history happened. Here's another view of the geological time scale, but this time accurately to scale. So if you take your hand and you put it out to the side, in fact, all the way from the tip of your, from the start of your fingers to your shoulder represents the Precambrian. And in fact, all nearly the entire history of multicellular life and animal life and plant life is contained in your fingers. But this other part of Earth's history, which represented by the Precambrian, it represents a microbial world, 3 billion years where microbes dominated the biosphere, and it's also a period when Earth saw some of its most dramatic environmental and chemical changes. So we're going to get right to the point. This graph right here shows you the concentrations of oxygen in the atmosphere over geological time from 4 billion years ago to today. And what you can see here, I'd like us to focus on the purple curve, which is the classic and long-standing view of the evolution of atmospheric oxygen concentrations. And what you can see here is that basically the history of oxygen in Earth's atmosphere is drawn in two large steps. The first step, around 2.5 billion years ago, we call this the Great Oxidation event. We went from an Earth that had very, very little oxygen, almost no free oxygen, to something around maybe 1/100th, 1/10th, and maybe even 1% of the current atmospheric oxygen level. This may not sound like a lot, but the jump was dramatic. Then, between about 2 billion years ago and about 700 million years ago, the Earth had this kind of intermediate oxygen concentration. It's not absent, but it's not as rich as today. And it's only in the last 600 to 450 million years, and this is a subject of ongoing research, that concentrations of oxygen in Earth's atmosphere reached near modern levels. So on this graph, you can see it's expressed on the right as partial pressure of O2, PAL, which means present atmospheric level. So what you can see here is that the oxygen in Earth's atmosphere has changed dramatically over geological time. What I want you to take home is that this happened in two large bursts, what we call the Great Oxidation Event, or GOE, and the Neoproterozoic Oxygenation Event, NOE. The reason why we say oxidation for the older one and oxygenation for the younger one is because in the GOE, we really went from a world that had no free oxygen to a little bit of free oxygen. And we saw oxidation for the first time of many chemical species and minerals. Whereas in the Neoproterozoic, we jumped from maybe 1% to about 100% of today's atmospheric oxygen level. So it's more of an increase in oxygen in Earth's surface. So that's why we call the Neoproterozoic event an oxygenation event, but we call the Great Oxidation event, we call oxidation. Now, how do we know this? We're going to spend the rest of this course really examining this curve and trying to understand in better detail how this oxygen accumulated in Earth's atmosphere. So we know this primarily by mineralogical and geochemical proxies. These are things that we can measure, chemical compositions, isotope compositions, or the presence absence of minerals that speaks to the presence of oxygen in the environment and in the atmosphere. So here is that same curve drawn again, but here you can see that we've divided various regions into regions that are compatible with the various proxies that have been used. These different proxies agree. We have some regions that are compatible with some proxies, but not others. And then we have in dark blue regions that are forbidden, that all the proxies eliminate this as a possibility. So you can see that we went from something vanishingly low prior to 2.5 billion years ago, and there was a 5 order of magnitude at minimum jump in atmospheric oxygen concentrations. The uncertainties around the actual atmospheric oxygen concentrations between 2.5 and about 0.5 billion years ago, there's large uncertainties, and it was likely dynamic in this time. And then you can see that we have the Neoproterozoic oxidation event capping this history around 500 million years ago. Now let's talk about these proxies in more detail. What are these proxies? Here at the bottom you can see a panel of light micrographs of minerals from a sandstone that dates before the Great Oxidation event. And the white minerals that you see, the white rounded grains that you see in the center of each of these micrographs, these are rounded pyrite grains. Now pyrite, you may be familiar with it, iron sulfide minerals. We find them often in organic rich fine-grained sediments that underwent anoxic diagenesis. The point is that today you would never find a sand with a pyrite grain in it, because these pyrites, they oxidize relatively rapidly. So if you go to a beach where you might have a cliff face with some sulfide-rich sediments, you'll see that it's actually rusting out, and you'll find it often full of holes, because sulfide minerals, like pyrite, are simply not stable in the presence of oxygen. They react with oxygen and they dissolve to sulfuric acid, and they're transported away. The fact that before the Great Oxidation event we find these minerals that are highly unstable in the presence of oxygen, we find them in sandstones, we find them in riverine deposits. This means that the oxygen concentrations were so low that we could effectively transport this mineral at our surface without dissolving it in the presence of oxygen. And this was one of the first lines of evidence used to really that really showed us that there was a dramatic change in oxygen levels in Earth's atmosphere sometime in the deep past. Other proxies are more sophisticated, and we're going to be talking about various proxies throughout this talk. Here, I'm not going to go into too much detail, but what you have here, you see geological time on one axis, and you see this strange triangle 33S. This is a capital delta isotope composition. So it's not a traditional little delta that you're used to, but it's a deviation from an expected value. It's an anomaly, and in this case, we're looking at anomalies on mass 33 sulfur isotopes. And today you can see that in the last 2 billion years these anomalies are non-existent. But prior to 2.5 billion years ago, when we find sedimentary sulfides, we often find very large anomalies specifically on the 33 isotope. And the only way that we really know to generate these anomalies efficiently is through some very special atmospheric photochemistry. Basically, we generate these anomalies by the interaction of intense UV light with SO2 in the atmosphere. So what this graphic is telling us is that before approximately 2.5 billion years ago, SO2 in Earth's atmosphere was subject to intense UV radiation, and after approximately 2.3 billion years ago, we don't see this photochemical processing of atmospheric sulfur. Why? It's because Earth developed its ozone layer, and it shielded atmospheric SO2 from UV light, solar UV radiation. So what you're seeing here, this is one of the most sophisticated proxies and is widely considered the smoking gun for atmospheric oxygenation. And as you can see, there are very few materials that deviate from zero after this Great Oxidation event. Essentially, the establishment of a little bit of oxygen in Earth's atmosphere and the establishment of an ozone layer completely cut off this unique photochemical processing that's specific to the early Earth. Now let's take a little bit of let's zoom in on this Great Oxidation event. Here you can see a zoom between 2.5 and 2 billion years ago. And what you can see is actually several different proxies listed on the right-hand side of this panel. So we're going to talk about some of these in more detail, but I just want you to see the overview of this transition and how multiple lines of evidence, multiple proxies line up to show this change in atmospheric oxygen at this time. So you can see that the first one is the presence of manganese oxide deposits, marine manganese oxide deposits. And manganese requires relatively high abundances of oxygen to oxidize and sediment out as manganese oxides. And even today in the ocean, in today's well-oxygenated oceans, a lot of the manganese is still present in reduced form because it doesn't oxidize even efficiently, even at today's atmospheric oxygen levels. So, well, before the Great Oxidation Event, before 2.5 billion years ago, we don't find these manganese oxide deposits. They're largely absent from the rock record, and we find the first ones around 2.25 to 2.4 billion years ago. You also see iron formations. These are deposits of iron oxides. We find a lot of them before the Great Oxidation Event, and they almost disappear after. And this is a sign, again, of increasing oxygen levels that are efficiently oxidizing iron and removing it from the ocean, such that we don't form these so-called iron formations anymore. So we see we have enough oxygen for the first time to oxidize manganese, and enough oxygen to actually probably largely remove iron from parts of the ocean. You can see the next item, the next proxy that is used to reconstruct this Great Oxidation Event are paleosols. What's a paleosol? Well, it's an ancient preserved weathering profile, an ancient soil. Not soils in the sense that you see around you today, but it could be a weathering profile in a granite, where the surface of the granite has been chemically altered, and as we go down in depth, this chemical alteration becomes progressively less until we reach unaltered rock. And by studying these alteration profiles, we can glean some information about the chemistry of the atmosphere and the presence of oxygen. And in the case of paleosols, before the Great Oxidation Event, there simply wasn't enough oxygen to oxidize all of the iron during weathering at our surface. And so the iron tended to be removed and mobilized from weathering horizons before the Great Oxidation Event. Afterwards, alteration profiles show an accumulation of iron that reflects this increase in atmospheric oxygen, the production of iron oxides, and the retention of iron oxides in soil like we see today. Then you see the next item down are red beds. These are essentially river bottoms that become oxidized. Prior to the Great Oxidation Event, whenever we find a riverbed, we don't tend to see an accumulation of iron oxides. Afterwards, we do, because atmospheric oxygen concentrations increased. You see the next item is detrital pyrite, which we talked about. We find pyrite grains rounded from their transport and sedimentary systems at Earth's surface prior to about 2.5, 2.4 billion years ago, and they disappear from the rock record for the rest of history for the large part. And there's another type of mineral proxy of this type, uranitite, which is similarly soluble only in the presence of oxygen. And they similarly disappear from the sedimentary record after the Great Oxidation Event. Then you see afterwards, we see marine sulfate evaporites. Today, sulfate is an important component of seawater, and when we evaporate seawater down to dryness, sulfate minerals form an important part of the solids that are released from this evaporation. However, prior to 2.5 billion years ago, sulfate minerals are largely absent from evaporitic sediments that result from the evaporation of seawater. And this is because the sulfate reservoir, the concentration of sulfate in seawater, was much lower prior to the Great Oxidation Event. Why? Because quite simply, sulfides, think about our detrital pyrites, they were not oxidized efficiently. So sulfur was largely trapped on land in reduced form, and it's only after an accumulation of some atmospheric oxygen that we could mobilize it efficiently to marine basins, and suddenly we find sulfate minerals as part of the evaporite record. We talked a little bit about the capital delta 33 record, this strange isotope-specific anomaly that is present prior to 2.5 billion years ago and absent after approximately 2.3 billion years ago. And there's another indicator, another sulfur isotope indicator that similarly speaks to oxygenation, but in a different way. And this is the more traditional small delta 34S, so the ratio of 34 to 32 sulfur, where these kinds of fractionations in the 34-32 system are largely generated by bacterial sulfate reduction. And the more sulfate we have, the more bacteria tend to fractionate isotopes of sulfur as they breathe this sulfate. Quite simply, before 2.5 billion years ago, there was very little sulfate in seawater, and bacteria did not fractionate these sulfur isotopes very efficiently. And we see the sulfur isotope fractionation in sedimentary sulfur minerals increase after the Great Oxidation event, reflecting this increased sulfate pool available for microbial processing that leaves a clear isotopic signature. There's a lot on this slide. We also see Earth's first, with a bit of a quotation mark, glaciation. It's rather surprising because the sun, as you'll hear in other lectures, was fainter on the early Earth. And one might expect that, that temperatures would have been colder on the planet. But in fact, the sedimentary rock record shows it's almost the opposite. We find almost no evidence for ice on the Earth in its first 2 billion years. There is one unit dated to about 2.96 billion years ago in South Africa that may or may not contain a glacial deposit. But other than that, there are no well-recognized or accepted glacial deposits prior to this Great Oxidation event. Why is atmospheric oxygenation causing glaciation? This is also subject to some debate, but prevailing thought is that prior to 2.5 billion years ago, in an oxygen-free atmosphere, we had a much larger methane component. And methane and oxygen are not necessarily stable in contact with each other. So some people, prevailing thought is that these glaciations were triggered by atmospheric oxygenation through the destruction of a methane greenhouse, or an atmosphere where methane played an important role in the greenhouse heating effect. And then finally, what you see on the very bottom, this curve is the carbon isotope composition of carbonate rocks. We're not going to get into detail on these in this lecture. However, it suffices to say that there were dramatic perturbations to the carbon cycle at this time. And one thing I'd really like you to keep in mind for later in the talk is what you can see labeled here as the Loma-Gundi event. And what you can see here is that sedimentary carbonates worldwide appear to have become isotopically much heavier, and for about 150 to 200 million years, and significantly heavier by about 10 per mil. This event, the Loma-Gundi event, represents Earth's largest and most long-lasting carbon isotope excursion. The relationship between this carbon cycle perturbation and atmospheric oxygenation is debated. But what is generally accepted is that this change in the carbon isotope composition of carbonate minerals represents an explosion of biospheric production. And as we'll see in the next couple slides, biospheric production can control atmospheric oxygen levels. Now, that was an overview, a very, I know, dense overview of this Great Oxidation event and this dramatic two-step history and that really important transition that was the Great Oxidation event about 2.5 to 2.4 billion years ago. Let's take a step back and ask ourselves, well, how do we oxygenate a planetary surface, an atmosphere? Well, there are three leading hypotheses. The first is something that we call hydrogen escape. And what you see here on this slide is it's a slide that shows on one axis thermal heating of various planetary bodies in our solar system, and on the x-axis we see the strength of gravity, which is related to the mass. Now, what you can see here is that there's a stark division. At high levels of heating but low gravity, we tend to find bodies that lack atmospheres. And why? This is because of thermal heating of any atmosphere that may have been present tends to excite the molecules and help them escape into space and overcome the gravity that's holding them to the planetary body. On the other end of the spectrum, at lower stellar heatings and higher gravity, planetary bodies retain their atmosphere. And what's quite important to note is that you can see here Earth and Mars, we're not very far from the line where at this scale, where we lose atmospheric components to space. Obviously, Earth still has its atmosphere, but Earth's atmosphere is leaky. So actually, we can zoom in a little bit more on this and ask ourselves which gases escape. So thermal heating excites gas molecules and helps them achieve the kinetic energy necessary for escape from gravity. But this process is different depending on the gases. So heavier gases are more easily retained, and in general, lighter gases escape more easily. And on Earth and on Mars, neither planet is capable of preventing escape of some of the lightest elements, notably hydrogen. And so this is actually kind of freaky. The image that you see on the right here is an image of Earth taken from very far away, looking at a hydrogen emission line. And what we're seeing, that red cloud around the Earth, is hydrogen gas bleeding off the top of the atmosphere and escaping into space. And so Earth is slowly losing its water to space, but not all of its water. What's happening is that water vapor in the atmosphere is being split into hydrogen and oxygen components. The hydrogen is light enough to escape, but the oxygen is heavy enough to be retained. And by this process of water vapor photolysis in the upper atmosphere and retention of oxygen and escape of hydrogen, this slowly leaves oxidizing potential behind the oxygen, while reducing power, the hydrogen escapes to space. And so this process, over billions of years, results in an accumulation of oxygen in Earth's atmosphere. Mars underwent a similar process, but because Mars is smaller, this happened much quicker. And in fact, the totality of Mars's oceans evaporated to space via hydrogen escape relatively early in Mars's history, and the result is we have a dry and very oxidized planetary surface. Earth is still undergoing hydrogen escape, and about 1 to 2 billion years from now, the fate of the Earth will be to become like Mars, dry and highly oxygenated. Potentially, that's one scenario. But the bottom line is this photo, which I find a particularly disturbing photo, is the Earth bleeding its oceans off to space progressively and oxidizing the planet in the process. Now, the calculations on the efficiency of this process suggest that it could account for the great oxidation event. The amount of hydrogen that modelers calculate we would have lost in the first 2 billion years of Earth's history is about the amount we need to oxidize Earth's surface reservoirs and leave oxygen, residual oxygen in the atmosphere. So this remains a very viable process for planetary oxidation. Now, it's not necessarily the preferred one for Earth's oxygenation, as we will see. The second hypothesis involves not producing oxygen, like the oxygen escape process, but it's actually about reducing oxygen consumption. And today, oxygen is produced and destroyed. And in fact, if we stopped all oxygen production on Earth right now, the atmosphere would tend down to zero oxygen within about 10,000 years or less. So oxygen is constantly being produced and it's being consumed by sinks. And one of these important sinks, is our reduced volcanic gases. Now, it has been proposed that the nature of these gases on Earth have changed through time. What you can see here on the left is this factor F, a fugacity factor. It's a measure of how oxidizing or reducing a gas is. And you can see here a comparison between gases issued from subaerial volcanism, that is, volcanism that occurs under the atmosphere, and it's compared to subaqueous volcanism, where we have volcanism and the escape of volcanic gases underwater. And what we can see is that there appears to be a fundamental difference in the oxidizing or reducing potential of these gases as a function of the source of these gases, whether they're being subaerial or subaqueous. Importantly, there seems to have been a dramatic transition in the nature of volcanism on Earth right around the same time as the Great Oxidation Event 2.5 billion years ago, where we went here on the graphic on the right, we're looking at the proportion, the subaerial proportion of lips. What's a lip? A lip is a large igneous province. It represents a very large eruption of lava at Earth's surface and a corresponding release of gas. And what we can see is around the Great Oxidation Event 2.5 billion years ago, there seems to have been a shift from more reducing gases issued from subaqueous volcanism to less reducing gases issued from subaerial volcanism. So perhaps a change in these volcanic gas sinks for oxygen may have contributed to that oxygenation of Earth's atmosphere. Finally, the third hypothesis, and I'd like to say that it is probably the leading hypothesis at this moment, and the most favorably viewed, is that the biology has had a very important role in the history of oxygen in Earth's atmosphere. And specifically, how can biology influence oxygen in Earth's atmosphere? Well, it's a bit more complicated than you think. We all know that plants can produce oxygen, algae and bacteria produce oxygen, but it's not sufficient to simply produce oxygen because we have to assure that it's not consumed. Think about a tree. Over the lifetime of a tree, it will undergo photosynthesis, take in CO2 to build the tree, and release oxygen proportionally. Now, if that tree dies, and oxygen is used to respire that organic matter and turn it back into CO2, there is no net gain. It takes just as much oxygen to consume the tree as the tree produced during its lifetime. So that net balance is zero. What really has to happen for biology to influence the redox state of the planet on a planetary scale is that we need to take that reducing power produced by biology. Biology is producing biomass and releasing oxygen. We have to take that biomass and sequester it, isolate it from the oxygen by burial. So by burying organic carbon, which is a strong reducing agent, by burying organic carbon and subducting it into the mantle, we leave excess O2 in Earth's atmosphere. And this is one of the leading hypotheses for the why and when of the Great Oxidation Event. And here you can see this is a nice photo of some colleagues visiting me in Brest, where we have some lovely, let's say, beachside geology. And here you can walk the entire Ordovician on the Brittany coast with beautiful exposure, and you can see these extremely dark black rocks. They're dark because of the organic matter they contain. And the amount of organic matter that we think has been buried over geological timescales is more than enough to account for the oxygen in Earth's atmosphere and the oxidation of all of the minerals at Earth's surface that accompanied it. Now, I'm going to focus more on the biology side for the rest of this talk. And what is quite clear, one of the reasons why the biology hypothesis is most favored is that there is clearly no metabolism on Earth that has made more of a significant impact than oxygenic photosynthesis that produces oxygen. We can see the consequences of oxygenic photosynthesis from space. Here you have the Bahamas carbonate platform. This is 7 kilometers thick of pure CaCO3 produced largely as a result of oxygenic photosynthesis. Not only does biology exert strong controls over the carbon cycle, like we see here through the production of carbonate minerals, but that all of that biological productivity and the organic matter contained in this platform equates directly to O2 equivalents released into the atmosphere. And so reconstructing the history of the biosphere is essential to understanding its role in Earth's surface oxygenation. But it's rather complex. So first off, did we have photosynthesis on the early Earth at the times that we're talking about? Was there photosynthesis before the Great Oxidation Event? Well, here you have a stromatolite, a calcareous structure that you'll hear about in other lectures that is produced by photosynthetic bacteria that are growing upwards toward the light, and their metabolic activity also triggers the precipitation of carbonate minerals, leading to these encrusted microbial structures called stromatolites. This one here is 2 billion years old, so after the Great Oxidation Event. However, we find stromatolites going back to some of Earth's earliest sedimentary records. Here is an overview of the diversity of stromatolites. As they take different forms, we can classify them like we classify animals. And you can see that stromatolites have had shifting diversity over geological time. The earliest records sedimentary records tend to contain rather simple forms of stromatolites, and they became very, very diverse in Earth's Middle Age, in the Proterozoic. They declined in modern times because the animal biosphere sees them as great food. And quite simply, the bacteria that produce these algal structures are eaten before they can generate these structures in most environments today. Now, this curve stops at 3 billion years, but our oldest well accepted bona fide stromatolites go as far back as approximately 3.5 billion years. So there is clearly this textural fossil evidence in the form of microbialites, specifically stromatolites, testifying that photosynthesis was occurring from about 3.5 to 2.5 billion years. There's a billion years of photosynthesis here. Now, that is not, sadly, it gets even more complicated because not all photosynthesis is the same. We are most familiar with what we've been talking about, oxygenic photosynthesis, the type that oxidizes oxygen in water to O2 using energy from the sun. But this is actually a very sophisticated form of photosynthesis, and there exist more primitive forms, what we call anoxygenic photosynthesis. And we're not making such a dramatic energetic leap from reduced oxygen in water to O2, but these are smaller steps. For example, oxidizing iron 2 to iron 3, which is a relatively small redox jump. And this is performed by bacteria containing what we consider more primitive photosystems that seem to be genetically ancestral to the photosystems used by oxidative photosynthesizers. So the problem is that we can't just look at a stromatolite and say, well, this is a photosynthetic structure, so it produced oxygen on the early Earth. Because this could have been formed by organisms performing photosynthesis without generating any oxygen at all. So this leads us to this idea that, well, how do we really know when biology started producing oxygen, well, free oxygen in great amounts, and when did the biosphere become very efficient at producing oxygen and burying organic carbon? Because these anoxygenic photosynthetic pathways are a little less efficient. We know of nothing on Earth today that is as important for the global carbon and oxygen cycle as oxygenic photosynthesis. So defining its first steps and its origins on Earth are crucial for understanding the role of biology in oxidation of Earth's atmosphere. So there are various ways to try to figure out what kind of bacteria were present and whether or not they were producing oxygen around the Great Oxidation Event or before the Great Oxidation Event. One technique involves what we call organic biomarkers. These are specific organic compounds. These are geologically resistant compounds that represent the degradation product of microbial biomass. And importantly, different kinds of bacteria can produce different markers that can be for which their degradation products can be analyzed in ancient rocks. And so it was proposed in the late 90s and early 2000s that there had been a successful detection of lipid biomarkers specific to oxygen-producing bacteria, cyanobacteria, at 2.7 billion years ago. So this would indicate that there were maybe bacteria producing oxygen before the Great Oxidation Event. Now, sadly, these results were controversial and revisited in a very careful and systematic way. And what you can see here are the analyses of two different kinds of biomarkers. But now we're seeing the analysis performed on the core outer surface, the core after contact with a saw, and then finally the inner part of this drill core that's been prepared under ultra clean conditions. And what we can see is that sadly these lipid biomarkers that were thought to indicate the presence of oxygen producing bacteria on the early Earth, we thought they were in this core. This isn't my work, but this is work from mostly workers in Australia and in the United States. They realized that in fact it appears that these biomarkers were modern contaminants, and that when they looked at the most pristine and carefully prepared inner parts of the drill core, the parts of the rock that had not seen drilling fluids or modern atmospheric contaminants, they don't find these biomarkers. And it's more of a question of preservation of these biomarkers. They're just not being preserved. Whether they were there or not, it doesn't seem like we can tell at present based on current technology. So sadly, these organic remnants don't seem to be really capable of telling us what kind of photosynthesis was occurring and whether or not the biosphere was producing free oxygen on the early Earth. A lot of the research has turned to other tracers of oxygen production on the early Earth. And this leads me to ask us to revisit this trajectory, this two-step trajectory of Earth evolution, not looking at the purple line, which is the classic view of a very simple two-step function, but the light blue lines, areas and arrows, represent updated, more nuanced view of how oxygen behaved in the Earth system on the early Earth. And what you can see here is that it doesn't seem that oxygen may not have just risen once and stayed there. There may have been an overshoot during the Great Oxidation event. As I had mentioned before, oxygen may have been very dynamic and may have descended by tens or hundreds during the Proterozoic, where you see that long, straight, shaded blue field. And that's a subject of active research. What I want to talk about right now are these little blue arrows, which are signs of oxygen before Earth's atmosphere became oxygenated. And so if we really want to understand what the role of biology is in planetary oxygenation on Earth, it's critical to understand, well, what are these traces, and what can they really tell us about whether or not there was biological oxygen production on the early Earth? Now, these arrows here, what we call these are pre-GOE pulses or whiffs of oxygen, as they're referred to. Here you can see several different paper titles that claim to find evidence for this early oxygen, and we're going to dive into a few in detail. I've co-authored some of these, and we're going to examine the evidence for this oxygen production with the goal, once again, of understanding whether or not the biosphere was capable of producing free oxygen for this first 2 billion years of Earth's history. So this idea that maybe biology was capable of producing oxygen prior to the Great Oxidation event and it was maybe present but cryptic, this idea was really launched by the work of Anbar and colleagues in 2007 in a seminal paper in Science called A Whiff of Oxygen Before the Great Oxidation Event. And this paper really was game-changing in that it made us realize that maybe there are small localized signals of oxygen production where the biosphere wasn't active enough to completely transition an atmosphere, but this metabolism may have been present on Earth. And the evidence for this whiff of oxygen before the Great Oxidation event comes in the form of trace elements. Here, what you're looking at here is a stratigraphic column of the Hammersley Basin, which is a sedimentary basin in Western Australia that is amongst one of the most well-preserved sedimentary basins that spans the time of the Great Oxidation event. And one of these units in particular, called the Mount McRae Shale, which has been dated to 2.501 billion years ago, contains tantalizing evidence of little tiny whiffs and pulses of oxygen before this dramatic, permanent secular change that was the Great Oxidation event. This evidence comes in the form of trace element enrichments, notably this little bump in molybdenum and a little bump in rhenium. Both of these elements are immobile in the absence of oxygen. Both tend to be found in sulfide minerals, and to liberate them, we have to oxidize some sulfide minerals, and we have to oxidize the molybdenum and the rhenium into oxyanions to transport them into seawater. To find these molybdenum and rhenium enrichments at this time reveals that there must have been a little bit of oxygenation, and that led to oxidation of sulfides and delivery of these oxygen indicators to this basin. It's notable that uranium does not show any enrichment. In this work, the authors claimed that, suggested that we'd reached a very small level of oxygen sufficient for oxidation of sulfides and release of molybdenum and rhenium, but insufficient for the oxidation of reduced uranium minerals. So we're at a really low intermediate redox state. This is still today rather controversial, and there has been other work. Here is an abstract from a Goldschmidt conference where Archean whiffs of oxygen go poof. And the authors of this work indicate significant open system post-depositional alteration of the rocks in question, and that particularly in the whiff interval there is widespread high temperature, late stage veining that may have generated signatures that aren't actually primary. Maybe these molybdenum rhenium enrichments don't actually date to 2.5 billion years ago, and maybe they're not recording oxygen. So this is rather controversial. Let's look at some other evidence for early oxygen production on the early Earth. And now we're going to go back to some of the most ancient evidence. This would represent one of the arrows at the farthest left of our O2 curve. And in this case, we're in South Africa, in the Pongola supergroup, and we're looking at some rocks that are 2.96 billion years old. In this sequence, we find some banded iron formations, carbonates, minor carbonate intervals, shales, etc. Now, what is remarkable in these 2.96 billion year old banded iron formations is a very interesting but sophisticated signal for the presence of free oxygen. And this is going to take a minute or two to explain. What you can see here on this graphic is the data from different iron formations of different ages, some deposited after the GOE, and some deposited before going back to 2.96 GA, the so-called Cinquenai iron formation in the Pongola supergroup. And what you're seeing on this graph is, on the y-axis, the ratio of iron to manganese on one side, and on the other axis we have the stable isotope composition of molybdenum. And you should recall that we mentioned how molybdenum requires oxygen to be liberated to seawater, but in fact there are certain steps in the molybdenum geochemical cycle that impart characteristic fractionations on the stable isotopes of molybdenum in the presence of oxygen. So to explain this briefly, here you can see on the very top, you can see that we have modern seawater hovering around a value of +2 per mil. So today molybdenum is isotopically heavy in seawater because minerals such as vernadite, as you see on the left, which is a manganese oxide mineral, removes molybdenum that is isotopically light. So here we have a kind of seesaw where the more manganese oxides we produce, the more they absorb molybdenum and they take out the isotopically light molybdenum, and this process leaves seawater enriched in residual molybdenum that is isotopically heavy. And so this light molybdenum that's being removed by manganese oxides, it's a testimony to the presence of manganese oxides. And the problem is manganese oxides aren't always preserved, especially in trace amounts, because they're a great thing to breathe on a very poorly oxygenated planet. So these rocks have no manganese oxides today. We don't expect manganese oxides to survive diagenesis and burial. They will be reduced to reduce manganese. However, what we see, this trend where as we go down to the far left corner to increasing manganese concentrations, we're finding isotopically lighter and lighter molybdenum isotope compositions. This is the telltale signature of the presence of manganese oxides in a rock where they wouldn't have been preserved. So this trend here towards this isotopically light molybdenum pole is evidence for the presence of manganese oxides as far back as 2.96 GA, which is in itself evidence for enough oxygen to produce manganese oxides. So this is one of the most ancient forms of evidence for oxygen on the early Earth. Now, there's also this work has also been proven controversial. In an accompanying commentary to this paper, it was stated that some of the samples come from areas where there are nearby intrusive sills that may have cooked the rocks, the sediments in question. Also, that maybe some of the outcrops were weathered. These were surface samples that, in the case of Africa, have been sitting at the surface for hundreds of thousands to millions of years. So this work is again controversial. And here's another paper claiming not just a little production of oxygen, but actual atmospheric oxygenation. It's a different thing to say there's oxygen in the atmosphere or there's just a little production, a local production and consumption. Here in this 2013 Nature paper, the authors claimed to find traces of free oxygen on the basis of chromium isotope fractionations and uranium enrichments. Now, I'm not going to go into the detail of these. You could look at this paper if you're very interested in chromium isotope tracing of redox. But the bottom line is that this has also been proven controversial because later authors went to the same outcrop, went to the same formations, but looked at drill core instead of surface samples, and the isotopic signatures reported in the Nature paper aren't found in fresh drill core. So this is highlighting the problem of finding a rock that might preserve a faint trace of oxygen 3 billion years ago, that is, but unfortunately maybe sitting at the surface for hundreds of thousands or millions of years on Earth's modern oxygenated surface. Now that's not to say that these are all controversial. Here we have a 2.8 billion year old carbonate platform in northern Canada called at Steep Rock, and here there is a very clear indication of free oxygen by the presence of a anomaly in the abundance of cerium, and we're going to talk about that in the last couple slides of this talk, about these cerium anomalies and this particular redox tracer. But here we have a case where our proxy is considered relatively robust. Rare earth elements are generally immobile and very difficult to reset by later alteration at their surface. And to find this particular signature at 2.8 billion years ago is a very strong evidence that there was production of oxygen at that time by the biosphere. There's also other tantalizing evidence for small amounts of oxygen being produced by the biosphere prior to the GOE. Here we have a compilation of sulfur abundances and molybdenum abundances in sedimentary rocks that show enrichments prior to the Great Oxidation event. And the best explanation for these sulfur and molybdenum enrichments are, once again, oxidative weathering on land, transport of sulfur and molybdenum to ocean basins, and here it appears to start as far back as 2.8 billion years ago in these compilations. So it is not all controversial. But now I think that for the last, so not all of these lines of evidence are considered controversial. Now we've already alluded to this, to a conundrum here that we have, where we have an anoxic atmosphere and potentially signs of production of trace amounts of oxygen on the early Earth. How do we reconcile this evidence for an anoxic atmosphere, but there are some tantalizing traces of free oxygen prior to 2.5 billion years ago? Well, one possibility is that we may have had what we call oxygen oases. The production of oxygen in upper ocean waters could have led to, according to some model calculations as presented in this paper, the accumulation of trace amounts of free O2 in the upper surface waters while the atmosphere remained completely anoxic. And so here is a model showing dissolved oxygen concentrations, which are still 1/50th of what we see in modern seawater, but non-negligible and sufficient to generate some of these signals that we're seeing for oxygen on the early Earth. So one possibility is that this is happening in the marine realm, where bacteria in the water column are generating sufficient oxygen to accumulate these little pockets of waters rich in oxygen. Some work I did with some colleagues looked more specifically at how these signals are generated. How do we oxidize these sulfides at what concentrations of oxygen and on what time scales? And what we're seeing here on this graph is atmospheric O2 in percent atmospheric level on the x-axis, and on the y-axis dissolution timescales of sulfide grains. And the bottom line is that at conditions, at atmospheric oxygen concentrations prior to the GOE, it's highly unlikely that we could alter sulfide minerals efficiently on rapid timescales. At very low oxygen, this process is slow. So slow, in fact, that we calculate in this paper that sulfide minerals will be buried and isolated from any potential oxygen oases in the marine realm before there is time to oxidize appreciable amounts of sulfide. However, on 10,000 to 100,000 year timescales, we could easily oxidize these sulfide minerals on land. And so here we suggested that, in fact, maybe we should be looking in terrestrial environments for these first traces of oxygen. And in fact, that's actually not really surprising when we look at the redox potential of natural waters across the Earth today. Here you have an EH-pH diagram showing oxidizing up high on the y-axis, reducing down low, and pH on the x. And what you can see here is that, in fact, surface waters rivers and groundwater occupy the most oxidizing end of the spectrum on Earth today. And that in fact marine waters, and especially marine pore waters, are not as oxidizing as we can find on land. So this is illustrated beautifully here with a piece of microbial mat. This is a photo of mine from Venezuela, where this microbial mat comes from a hypersaline lagoon where there are stromatolites. Here's a photo of that lagoon, and the mat comes from the surface. But what's really incredible here is that you see those bumps on the surface. Those are bubbles of pure O2. Here you can see the graph on the left shows microelectrode profiles, so at very, very small scales, showing oxygen and sulfide concentrations within these microbial mats. And in fact, in that upper, very active photosynthetic layer, we have nearly pure oxygen in the top. And it turns out that in fact, O2 production on Earth is highly compartmentalized. And we don't find it in, nowhere do we find it in greater abundance than in benthic or surface-attached microbial ecosystems. And in fact, oxygenic photosynthetic rates in microbial mats can be 1,000 to 10,000 fold greater by volume than communities living in a water column in the ocean. So in other words, 1 centimeter microbial mat, one centimeter thickness can produce as much oxygen as a 100-meter thick photic zone of an ocean water column. So here are a couple examples of modern oxygen oases today. Microbial mats, as we mentioned, produce oxygen. Here we have a hypersaline lagoon where you can see microbial mats desiccating and contracting away from the underlying sand. On the right, you have hydrothermal microbial mats developed in a hydrothermal setting. And these are very oxygen-rich ecosystems. Also, soil crusts and endolithic bacteria that like to live inside rocks, tend to generate large amounts of free O2. Riverbeds and estuaries are net sources of O2 to the atmosphere as well today. So some work by myself and colleagues has looked at these oxygen production rates in different environments. And we can kind of establish a net O2 production rate as a function of surface colonized. What gets really interesting is when you extrapolate this oxygen production rate by microbial mat communities to large parts of the continents, it turns out that you only need small degrees of continental coverage, what you see down here on the right on the x-axis, to generate oxygen equivalent fluxes that are easily sufficient to account for this sulfur fluxes that we saw 2.8 billion years ago, or to account for other important steps in the oxidation of the Earth's surface system. So the bottom line is that production of oxygen may have occurred on the early Earth. The earliest production may have been happening in very small, in surface-bound ecosystems where oxygen wasn't necessarily escaping the atmosphere. It was being produced and consumed, but if part of that oxygen is shunted into oxidizing some minerals, we would expect to find the signals that we're seeing for oxygen in an otherwise anoxic world. Now, this isn't so clear-cut because in reality, when we look at certain microbial mats, especially those that are more analogous to what we would expect for the early Earth, oxygenic photosynthesis doesn't happen all day long. But in fact, there's competition between different kinds of photosynthesizers, and the earlier work on these oxygen production rates of these benthic microbial ecosystems may have overestimated their oxygen-producing capacity. And in fact, certain modern mats produce less oxygen. Now, finally, I'm kind of going to throw out, this is a bit of speculation now. If cyanobacteria were locally producing oxygen before the GOE, they were present, well, what controlled their rise to prominence? Why were they producing relatively little, and then they eventually may have produced enough to oxidize the planet? Well, this may be related to the amount of continental surface the early Earth had. On the early Earth, we didn't have the same area of continents as we have today. And in fact, prevailing models suggest that the early Earth was a near water world prior to 3 to 3.5 billion years ago. So there may have been actually very little continental surface for these highly efficient oxidizing microbial communities to inhabit. So we're gonna end on a little study of one of the earliest known communities living in this kind of a context where we have a terrestrial and a very niche nearshore shallow water habitat. So this is just we're going to end this lecture on a very short example of looking for a signal of oxygen at 3.2 billion years ago. So a little bit further than most studies have looked, and we're going to see some promises and pitfalls when we go chasing some of these communities that may be most likely to produce these first traces of oxygen on the planet. So we're going to go to the 3.22 billion year old Moody's group of the Barberton Greenstone Belt in South Africa. So here on this map you can see the little inset at the bottom right of the map shows where we are in South Africa. So we're near the frontier with the border with Swaziland, and here you can see on the right in yellow, light green, and dark green a simplified stratigraphic column of the Barberton Greenstone Belt, where we have the Omphurvat group, the Fig Tree group, and the uppermost Moody's group, the latter being mostly composed of sandstones, deltaic nearshore sediments, even a riverbed system that is preserved, and importantly, some iron formations. Now, here are some of the sandstones of the 3.2 billion year old Moody's group, and what you see here in black has been interpreted as compressed microbial mats. And so this is generally accepted. There is some controversy, but this is a spectacular preservation of microbial mats growing on sand, if that's what it truly represents. The problem is, so how would we go about looking to see if these early communities were producing oxygen? Well, is there any traces of oxygen in these mats? Sadly, in a sandstone, there's relatively little to analyze that tells us about the chemical environment. This is largely a physical product of weathering, and there's very little chemical signatures preserved in this kind of a rock. However, luckily, in the Moody's group, we have several very shallow banded iron formations. Banded iron formations are chemical iron-rich sediments that are really good tracers of their chemical environment. They precipitate directly from seawater, and they capture chemical signatures of that water when they precipitate. Now, I'm going to show these last couple slides. We'll look at the rare earth element systematics of these shallow water banded iron formations at 3.2 billion years ago. The rare earth elements, as I already mentioned, are particularly attractive because they are relatively immobile. They're a lot more difficult to mobilize with or without oxygen by any waters due to their general insolubility. And what is also very nice about the rare earth elements is that they contain characteristic signatures in their rare earth element patterns that tells us about things like whether oxygen was present or not, whether or not this is a marine sediment, because seawater has a certain signature. So what you can see in this graphic here are, on the upper panel on the left, different, in white, different oxygenated ocean basins today, where you can see that modern seawater is characterized by a general heavy rare earth element enrichment. So that means we have on the right, we're slightly higher in rare earth elements than on the left. We see a positive anomaly in lanthanum at the far left, and a positive yttrium-holmium ratio. These are all characteristics of seawater that we find going back to Earth's oldest marine deposits. So here we have a double check that this is a marine deposit. And then you'll also notice in black we have two modern anoxic basins. And there's an important difference to note here is that in the anoxic basins you can see that cerium behaves like its neighbors, with the exception of lanthanum. However, when we look at oxygenated waters, we see that they are remarkably cerium poor. This is because cerium is unique amongst the rare earth elements in its reactivity with oxygen. And so cerium 3+, in the presence of oxygen, oxidizes to cerium 4+, which is even more insoluble, and it gets removed from natural waters relative to its neighbors because of this oxidation. And you can see on the bottom, you can see some iron formations and carbonate platforms of different ages, where in black, sediments deposited just before the Great Oxidation event don't have this cerium anomaly. And then there's some modern carbonates that do have this cerium anomaly. And here you can see cerium anomalies that are growing progressively bigger and bigger, so going from 1, which is no cerium anomaly, the same amount of cerium as the neighbors, going down and down and down to 0.2. And here you can see what you're basically seeing from the cryogenians, so from about 700 million years ago. You're seeing these cerium anomalies getting more and more important in marine deposits as the system became progressively more and more oxygenated at the end of the Neoproterozoic. Now, the surprising thing about the Moody's group, when we look at clastic rocks like the sandstones, we get a flat signature that represents the continental crust, and we tend to show these signatures in by what we call shale normalization. We normalize the concentrations measured in the rock to a shale composite that represents the upper continental crust. And there's no surprise here, the sandstone resembles generally the upper continental crust in its rare earth element signature. However, when we look at the Moody's group iron formations, we find something very different from the upper continental crust. We find a seawater signature. So the Moody's group is on the bottom, where you have a positive lanthanum anomaly, you have a positive yttrium-holmium ratio. This europium anomaly is that in the middle there is indicator of high temperature hydrothermal fluids contributing to the deposit. And what you see in the box above is a 3.8 billion year old banded iron formation from Greenland, which is actually one of our geochemical reference materials, a geological reference material that shows a very similar spectra. So here you're looking at seawater, 3.8 billion years ago and 3.2 billion years ago. This is what some of the Moody's BIF look like. Here's some others. And right away, you might notice this striking cerium anomaly. So we were very excited. Oh, maybe we have evidence for oxygen production exactly where we think it should be, these benthic microbial communities at 3.2 billion years ago. Now, here is these cerium anomalies expressed. Here you can see them getting more and more important as we go to the lower left of the graphic. And in blue is modern seawater. So you can see that, wow, this almost looks like we have modern seawater like oxygen levels 3.2 billion years ago. This was really exciting when we saw this data at first. We were very concerned about the idea of alteration. So we did some geochronological dating. Here you have samarium neodymium isochron. So this is basically a samarium neodymium dating technique where we have these banded iron formations are in red, and the white points are other data from across the Barberton Greenstone belt. And what you can see here is that we get a samarium neodymium age of within error of the known depositional age of 3.2 billion years ago. So, well, this is already a good sign. We're actually dating some rare earth elements that are right next to our oxygen signal. So this would at first glance indicate that maybe these rare earth elements are pristine and haven't been altered. But however, it turns out that, well, samarium and neodymium are actually very difficult to separate from each other because they behave chemically similarly and neither one is sensitive to oxygen. However, this sensitivity of cerium to oxygen changes the game a little bit. We also performed we performed lanthanum cerium geochronology, and this is a difficult dating technique. What you're seeing here is the first sedimentary lanthanum cerium isochron produced by Pierre Bonon at Clermont-Ferrand, and what we're seeing here is that here we have an age that's actually only 60 million years old. This is very confusing. What we have on one hand, we have a cerium anomaly where the cerium itself is giving a 60 million year old age, but its neighbors samarium and neodymium are giving 3.2 billion year old ages. What has happened here, we determined, was that there was later perturbation of this system, but this perturbation affected mostly cerium, because it can be separated from its neighbors by oxidative processes, whereas samarium and neodymium were unaffected by this alteration process. So this is a word of caution. We don't believe there was any oxygen in this deposit. It's that this kind of illustrates the difficulty of finding trace amounts of oxygen and being certain that they come from the ancient biosphere and not necessarily from modern weathering. So this is a major issue right now in this kind of research to better understand when did the biosphere first start producing oxygen and how did that contribute to Earth's oxidation history. It's something that is rather difficult to do because we are awash in oxygen today and a lot of these rocks have seen that oxygen. So in conclusion, Earth's surface oxygenation proceeded in two major steps: the Great Oxidation Event and the Neoproterozoic Oxidation Event. And there are three popular hypotheses for what drove Earth's surface oxygenation: the escape of hydrogen to space, leaving oxygen behind, volcanic gases that became less reducing and thus less oxygen consuming over geological time. And then finally, biospheric effects like carbon burial and the production of oxygen and organic matter by oxygenic photosynthesis. We now believe that highly localized oxygen production was likely highly important before the GOE for Earth's surface oxygen cycle. In some cases, even the most robust proxies for O2, such as the highly immobile rare earth elements, are susceptible to being reset by later alteration processes. So we have to be very careful about what we're analyzing and what we read in the literature, because this problem may be more widespread than we thought. And if we're really going to get to the heart of when and how the biosphere affected Earth's oxygenation, the early Earth's oxygenation, it would be great to have some geochronologically resolved bulletproof signal for free O2 at that time. So with that, I hope that you've learned new things about Earth's early oxygenation, and feel free to contact me if you have questions. Thank you.